So again, this is chapter 10 on how cells divide, if this wants to work.
Do you need to do the mouse next?
Yeah, it's cool. So one of the things we did not talk about last time was last time we looked at what kind of cells. Were they prokaryotic or eukaryotic cells? Wait, let's slow this one down. Okay. We looked at plants. We looked at plants and we looked at animals. And so they're eukaryotic. We know those are eukaryotic. Okay? We know those are eukaryotic because they had a nucleus, right? Because again, if you break down that word, if you break down prokaryotic and eukaryotic, pro means what? No. Well, it sounds like no, but it actually means before. And karyos, the Greek word karyos, K-A-R-O-S, means nucleus or center. So it's before nucleus, and then eu actually means true. So eukaryotes have a true nucleus, whereas prokaryotes are before the nucleus. Okay? So most prokaryotes, like bacteria, don't go through the full steps of mitosis. Because they don't have a nucleus, because they don't have a lot of DNA, because they can very easily replicate that DNA, all they do is they almost do like a magic trick. I don't know, it kind of reminds me of a magic trick. You ever seen the magic trick where the two rings? And he goes 1, 2, 3, and he breaks them and puts them together. You're like, look, we're all together. And then he goes 1, 2, 3, and spins them, and they come apart. Right? It's the same kind of idea. So if you had two circles, okay, you can only see one circle right now. And basically what happens is as the DNA of the bacteria replicates, as it copies itself, and again, it should be pretty much exactly a copy. There is two sites you need to know about. There's this green site, which is known as the origin of replication. Right? Here. It's the very top. Y'all don't have green, obviously. But it's the very top. This picture is from your book. That is a reverse picture. So it's known as the origin of replication. Okay? It's called the origin of replication. So this is where replication starts. So the DNA is starting to be fed, or starting to be read from that origin. Origin means start. So it's the replication starts at the origin of replication. And basically what it does is as it's being fed, as it's being read and more DNA is being made, those two little circles, all of a sudden you start seeing a separate circle start pulling out. And eventually you get two circles, they actually break apart, and you get two circular DNA. Then those two separate out, and you have now two complete DNA strings. Okay? So prokaryotic DNA is not linear. Our DNA, eukaryotic DNA, is linear. Right? So when y'all were freshmen, y'all made the little model thing, right? The little DNA model? Like that's what's up in the room? Okay? No? Okay. Well, last year my freshmen made the DNA model. They'll probably make another one this year. That's what the gigantic doorstop is outside. The big metal one. Okay? That was the one my freshmen made last year. That twisted, that double helix is linear. Okay? And your DNA is always is linear. It's not circular. Because it has a start and an end. Okay? And that DNA is wrapped around itself and bundled together and wrapped together and bundled together and all this stuff. Whereas prokaryotic DNA is circular. Okay? And sometimes the prokaryotic DNA actually has small parts. And we'll talk about those small little, what are they called, plasmids. And we actually use plasmids to make drugs and insulin and all kinds of different other fun medical type things. Okay? So prokaryotic DNA is circular, and so that's the reason why it goes through these steps like this. So it goes through this idea where basically the origin of replication goes through and starts doubling off. And then this is the ending strand, that little red part. That's called the termination site. So the termination site is where it will actually finally pinch off. Okay. Now, there's not much else going on. The only thing really that's going on here is that then the cytoplasm basically doubles and it splits off. Okay?
So, you just have two things in DNA and they're not close to anything, they're just like...
They're just floating up. John, do I need a hammer?
So,
Right? Now,
does it not have a start and an end because of that termination site?
yeah, so it doesn't. So the termination site is the ending site, and the origin replication is the beginning site. And it actually feeds backwards. It actually feeds forwards and backwards.
Wait, what?
Okay, so it's a circle, right? And like my pinkies are the origin site. So it's going to feed this way, and it's going to replicate this way as well. So, but
So it's like replicating, making a new circle both ways.
yes,
And then the new circle goes.
yes. So like, and it always replicates from 3' to 5', which we'll talk about that later. Don't worry about that. But it always replicates in that order. And so this order is the correct order. This is the incorrect order. But it finds ways to fix that. And we'll talk about those fixing ways later. But what you need to know right now is that it doesn't go through mitosis. It doesn't have to go through the metaphase on the chromosomes line up and phase and
So
hold
it's
up.
replicating on the back of the already.
Yes. Yeah. Yeah.
And the red is the finishing.
Yeah. It's called the termination site.
Because it finishes.
That's where it finishes. And so my thumbs will be the termination site. So when this one feeds all the way to the thumb and this one feeds all the way to the thumb, then it basically flips over. Okay, the little rings flip over. And now you have two. Those separate out. Okay, and then some cytoplasm comes with this one, some cytoplasm comes with this one, and then in the middle, it's called the septum, and the septum is where the two are going to pinch off.
So
Okay.
do the half circles make circles?
That-
Like when they break apart?
These half circles right here. This one.
Yes.
Basically, what's happening is that this one, it's hard to show. Actually, let me show you the animation. But basically, what's going on is this other circle basically is slowly, because they're feeding from both sides, it's basically making it bigger and bigger and bigger.
Okay.
Right? And so once it's all the way the same size as this, it then separates out. It then completely conjoins. It breaks apart, and new cell walls form. Now you have two exactly identical bacteria.
So do these cells have to grow as they do that?
Yeah, they're getting bigger as they grow.
So where do they get that?
They actually just double their cytoplasm. They just go through the same process of protein. You know, basically before it goes to this, it's going to be able to have an increase of ribosomes. It's going to have an increase of cytoplasm. It's going to have and it's going to go through each of those steps.
So it's kind of like
It prepares before it does this. Okay. There is a little animation I want to show y'all, kind of what this looks like. Eventually.
that's redirect.
So like I said, I'll come back to the animation. I'll pull it up once it finally loads. It's loading on my other side. So once it loads, you do need to understand a couple of vocabulary terms. Again, these are the vocabulary terms on your list. So refresher, what's a somatic cell?
Isn't that a cell like a sperm?
Yeah. It's any cell that's not a sperm or an egg cell. Any cell, from your head to your toe, is a somatic cell, except for your gametes. And so then your gametes, that's the next one, is then, if it's not a sperm and egg cell, then the gamete has to be the sperm or this egg cell. Okay? So any cell, your toe cell, your hair cell, your eye cell, your booger cell, whatever it is a somatic cell. Okay?
Every cell's a somatic cell?
Every cell in your body, other than your gametes, your sperm and your egg cells, are somatic cells. Okay? So, yes, your egg cells too, Mason. But now here's the thing, though. If you get cancer, if you get skin cancer, can you pass that skin cancer on to your kids?
No.
No. Okay? But if you get cancer within your gametes, those will be passed on to your kids. That mutation, okay, that mutation within the gonads can then be transferred on to your kids. Okay? Because only the gamete cells are passed on to your offspring, not the somatic cells. The gamete cells make new somatic cells, but only mutations within those cells is when they're then going to be passed on.
So you could only...
Haploid and diploid. What does di mean? What's the prefix di? So 2. So that basically means you have 2 copies of every single chromosome. Okay, that's known as 2N. If N is the number of chromosomes you should have in your body, then 2N would then be the diploid number. So if you're supposed to have 23 chromosomes in your body, that's your haploid number. So what's your diploid number? 46. Okay, that's how many chromosomes we have in our body. Now, just because the organism has less chromosomes than that doesn't mean it's a lower organism. And just because it has more chromosomes than that doesn't mean it's a greater organism. For example, there is I believe a species of plant that has something of the number of 46 haploid chromosomes. So its diploid number is 92. Okay? They're not as specialized as we are, but yet they've got more chromosomes. Okay? There are certain mammals that only have 8 chromosomes. There are certain mosquitoes that have 16 chromosomes. Okay? So just because the number of chromosomes doesn't determine its hierarchy, its order in the greatness of life. Okay? The centromere, again, this is that part that's right here in the middle. This is what is going to connect to those spindle fibers. Okay? So the centromere is the middle. The kinetochore is actually the part that connects to the spindle fiber. So the centromere is a little pinch in the middle. Kinetochore is the middle. Each of these, so this would be like your daddy chromosome. This is like chromosome number 1 from your daddy. This is chromosome number 2, or chromosome number 1 from your mommy. Okay? You get both copies. And so every single little different thing about you is because of the different types of chromosomes from you. From your different parents. So daddy might have said that you have to be really tall. But mommy says that you have to be really short. So what are you?
Medium.
You're actually medium height. Okay? And again, we're not going to genetics right now. We'll go to genetics, I think, next unit. But it's from both copies. So when you look at mitosis, you're not that, let's say we've got 4 chromosomes. When you go through mitosis, okay, you're going to have homolog chromosomes on both sides. So you're actually going to have, it's just random as far as it's going to actually copy both the mommy and the daddy chromosome, okay? And that's going to be separated out on both sides. So you're going to get your mommy chromosome, that's going to make another copy of that mommy chromosome. And if you have your daddy chromosome, it's going to make another copy of that daddy chromosome, okay? And both the mommy and the daddy chromosomes are going to go to one side, and both the mommy and the daddy chromosomes are going to go to the other side, turning in a face. Okay? So you get both copies every single time because you're making an identical copy. Because if not, if you're going through the process of meiosis, you get a copy of mommy and a copy of daddy, and then there might be a chance that this cell gets a copy of daddy's, and then this cell gets a copy of mommy's. Okay? I'm being really, formal words here. Okay, so those are called chromosomes. These are called homologous chromosomes. This is both from chromosome number 1 or number 5 or whatever it is. Okay, those would be homolog chromosomes. Then sister chromatids would then be, once we go through replication, okay, this is in the S phase. Once we go through replication, we're going to make sister chromatids. These are exact copies of the same chromosome. This is chromosome number 1 from dad. This is chromosome number 1 from mom. But your sister chromatids are the same, is the copied form of the same chromosome. Okay? Let me repeat this one. So, your sister chromatids, okay, is all it is basically if you had a chromosome, it's going to go through replication. Replication is a copy of DNA, exact copy of DNA. That one chromosome is going to make 2 chromosomes, 2 exact pairs. Okay? And the reason why it has 2 exact pairs is because at the end of mitosis, how many cells are you going to end up with? If you start with 1, how many are you going to end up with? 2. They're going to be exactly the same. So every single cell, both the cell that was the original form and the new daughter cell, has to be exactly the same. And so if you've got a chromosome, then there needs to be 2 copies of that chromosome. But remember, you get a copy from each parent. So you get a copy. You have to make a copy of the dad chromosome and a copy of the mom chromosome. Okay? So when you go through mitosis, not only are you separating out the mom and dad chromosome, you're separating out 1 chromosome from the mom and 1 chromosome from the dad. Okay? So the sister chromatids is when they've been copied. When you go through DNA replication and both DNA split apart.
So it's the same, like the blue are the exact same chromosomes.
Yes.
Made like twice, basically, or one's
Once,
copied.
copy, yeah.
Combined.
Yeah. Okay? It's like this idea that if I have to give, if I've got my copy of a paper, okay, of a test, and I have to give that same test to Miss Morgan because she's going to give it to her class, okay, I have to run a copy of that test to then give to her. Okay? But now let's say we've got part A and part B. So I have to run a copy of part A and a copy of part B, and then I have to give both A and B to her. But I keep the original. Yeah. Does that make sense, that whole idea?
Oh, so the parents are giving copies of theirs
Yes.
to the.
It's not an offspring, it's the new cell.
Oh, sorry.
So what you're saying is you have one original cell, and it copies its DNA, and makes another DNA, and it splits into a different cell. But the DNA that it replicated is called its sister's DNA, and the new cell is its daughter.
The new cell's daughter, which is really weird, right?
So your daughter,
The daughter's cell
sister.
contains a sister chromatid. And my uncle is my father's mother.
Mom, dad, sister's daughter.
Okay?
My uncle was.
Don't worry about it. So again, this is the idea that if this part right here, this little bitty part right here, and this graphic isn't actually very clear because from G1 all the way to G2 is about 90 to 95% of the time. Okay? This is about 5 to 10% of the time. So this purple and this blue should actually be like maybe as much as the blue. But they had to break it apart. Okay? And this is where you find prophase, prometaphase, metaphase, anaphase, telophase. So interphase, I, P on the mats. I don't know.
What? PP math?
Yeah. So interphase, I, prophase P, metaphase P, metaphase M, A, T. So I, P, P on mat. Just go with it. It's cool. Now there are very specific ideas within each of these. Okay, but we're going to look at these when we play the game today. So hopefully after playing the game you'll understand what goes on during the G1, S, and G2, as well as during the M phase. And M phase does include both mitosis and cytokinesis.
Wait, cytokinesis is in the M phase?
Cytokinesis is at the end of the M phase. It's within the M phase, and it's at the very end of mitosis. Okay, so we went through each of these. I don't think I really need to go back over these, do I? So prophase, basically the nuclear envelope disappears. The centrioles start sending out spindle fibers. Those are then connecting to the chromosomes, and that's what's going to actually help move it around. And so then prometaphase is basically when the entire nuclear envelope is gone and the spindle fibers actually have been attached. Okay, so prophase is basically you start seeing the nuclear envelope going away, and you start seeing the spindle fibers coming out, but they're not fully attached yet. Whereas in prometaphase, you will truly see an attachment. And then you go into metaphase. That's where the chromosomes line up on the plate. I tried to give you a picture of both the animal cell as well as the plant cell. You can't really tell the difference right now. Okay, but again, it lines up along the metaphase plate, and that metaphase plate is perpendicular to the centrioles. So the centrioles are right here and right here. Okay, can you all see the spindle fibers now from the centrioles? Okay, so that metaphase plate is perpendicular. So this is the centriole line. That metaphase plate is perpendicular to that. That's why they're all lined up on that line. Okay, and this is where cleavage will occur. This is where that new plant cell wall will occur as well, because of the pinching. And the anaphase is where it's actually being pulled apart. Okay. This is actually the really cool looking one because you see the chromosomes actually moving. They do move to the opposite poles towards the centrioles. And telophase is where nuclear membranes are bound both sides of the chromosome, and nucleoli reappear. That nuclear envelope reappears. And again, I really like that picture right there because you can start to see that cell plate forming. Okay. This again is the furrow coming in. Cross your fingers, see if this works. So this is 2 hours broken down, 1 hour broken down into about 15 seconds.
Differential interference contrast spectroscopy is used here to visualize mitotic events in a lung cell growth tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
So, do all cells happen in an hour? Or
No.
do you-
No.
different?
So like your skin cells take about 24 hours. That was a newt. That was a lung cell of a newt. So it's again, so newt's very, very short. But then like your liver cell actually takes years. Bacterial cells that go through binary fission takes a couple hours, an hour if that. Each cell has a different time frame, and basically based on how intense it is, how many different ways they have to fix the replication, those kind of things. How quickly is it? So like your skin cells, it takes about 24 hours because you're constantly renewing your skin cells. Your liver cells, you don't really have to because you basically just continue to use them. So it's based on how basically the frequency of use, essentially, the frequency of degradation or that breakdown of them. So I always like to turn down the volume and see if you can kind of point out the different steps. So what step is this? It's interphase. Good. What step is this? Pro. And then what step is that? Pro-meta. And then what step is that? Metaphase. Can you all see that line? How they're all in the middle? Obviously now you can see the line. And this is anaphase. And then next is telophase. Forms inwards. Again, these notes are online, so you can watch this video, and I'll also put the link down below too. So I don't know if y'all can read that or not, but just there.
So prometaphase is just them like having the chromosomes like lining up in the middle.
Prometaphase is where you will truly see the nuclear envelope disappearing. You'll see the spindle fibers actually attaching to the chromosome, those kind of things. So this kind of breaks down what the mitotic spindles are, but just know that they are microtubules and proteins. They do have those that connect to the chromosomes and those that do not. And it's just the kinetic core microtubules and those are non-kinetic core microtubules. And they both basically have the same effect of moving the chromosomes around as needed. So here's a couple pictures from the book itself. There is this adhesion protein right here. And if that cohesion protein doesn't exist, then what could happen during mitosis? If let's say that cohesion protein broke down and it's moving the chromosomes around, would those chromosomes be split up properly? No. Okay. If you're in a busy subway of like say Grand Central Station of New York, and you got like when we go to New York this Thanksgiving, I'm gonna have to hold on to my daughter, right? So make sure that we get to the right subway or whatever. But if she loses grip and she goes on, she could end up somewhere else. She could end up in the Bronx, or she could end up in Jersey, and that just would be bad, right? So we have to make sure that cohesion, that protein cohesion stays together, because as the kinetic cores are moving the chromosomes around, moving towards the middle, pulling them apart, that protein needs to stick together until the part of anaphase and when they're actually ripped apart. Here's a couple of different pictures of the different cytokinesis. Again, plant cell walls. This was a question I had in another period. Those cell walls, actually what they do is they actually form holes within the cytoplasm itself. Those holes are actually covered in cellulose, and that cellulose will then become the new cell wall. Okay. So, question. What phase is, let's say that one. I would say pro-meta, yeah. What phase is that one?
That's
Anaphase. What phase is this one?
Which one?
These ones, like all this whole thing right here.
Prophase.
Nope.
Is that
Telophase, yeah, because this is right after it's done. What phase is that one right there? The chromosomes are starting to be seen. The nuclear envelope is starting to go away. After interphase. Prophase. Interphase would be basically any of these ones you can truly see the nucleus, like that one and that one and that one. Trying to find any good ones I missed. Metaphase is really kind of hard to see in this. I think that's more of a metaphase. Like you might even call that one metaphase. I know we call that prometaphase. But
That one looks more pro.
this one?
No, other
This
right
one?
next to it.
This one?
Yeah.
Yeah.
That one looks more.
Yeah.
that one.
Okay. So make sure you can do that. This is the picture just from the book. I really like how they... oh, this is not that picture. This actually is a picture. Don't worry about this one, but this is actually a picture about how we know... the book goes really in-depth into this, what's called the FTS-Z protein. I don't think you really need to understand that, and I'm not going to test you on that idea. But basically the idea is that it's this whole evolutionary idea that basically the same ideas that go on in prokaryotes kind of last through protists, through yeast, and then finally into animals. This idea where there's this polar microtubule and how there's a certain center area protein, and that protein actually determines where certain things split. There are certain controls. This is the part that we haven't really covered yet. There are checkpoints. Okay, there are very specific checkpoints. There's a G1 checkpoint, a G2, and an M checkpoint. And each of these are broken down up here. Y'all can read those. But the main thing that is actually that's actually controlling these checkpoints is something called cyclin. And cyclin is a protein. Cyclin is actually an enzyme, and it actually has two different places it can be phosphorylated. So it can be phosphorylated in this area. And this is actually the cyclin-dependent kinase, which is an enzyme. And if it's phosphorylated one way, it actually allows the cyclin to be in here, and it actually inactivates it. So let's say that you've gone through and you replicated all your DNA. Okay, we've replicated our DNA, and the cell is checking the DNA, and they find a mistake. What they can do is this cyclin will actually phosphorylate and stop that continuing on, right here in G2. So it's checked for replication. It found a mistake in the replication. It'll actually stop right there in G2, and it'll go back and fix that. And so it'll actually phosphorylate with the cyclin attached to it. Okay, but let's say it goes through, it replicates, everything's good. It's going to go up here to mitosis. It passes this checkpoint. It's actually going to then phosphorylate on the other side, and that's actually going to remove the cyclin. Okay, and just dependent upon where the phosphate is, as dependent upon where... if it's activating or inactivating. The APC is just another type, so cyclin, CDC2, and APC are all types of checkpoints, basically determining if it can continue on. The reason for checkpoints is things like cells, or things like cancer. Okay, cancer is where there is the checkpoints, checkpoints are basically just blown past. Okay, so that DNA has been synthesized. It's got a mistake, but it's a cancer cell. So even though it's got a mistake, it blows past that first check. It blows past that second checkpoint on G2, and it continues to replicate. It goes through mitosis, and the cell can find that that mitosis didn't work exactly right, but it blows past that checkpoint. And it continues to do that over and over and again. And so, what this picture is talking about is this P53, which is a protein, and basically, if the protein exists, then it'll go back and repair this enzyme. It'll repair this DNA. But if the protein is not there, it'll have that abnormal cell. That abnormal cell will go through mitosis, make another abnormal cell, make another abnormal cell, and eventually these will become cancer cells. And all cancer really is just an unregulated cell growth. So if you go back to this cell cycle here, okay, this whole cell cycle is unregulated. There are no checkpoints on cancer cells. They just continually grow. Okay? That's the reason why you always they say check yourself for skin cancer, and the easiest way to is the ABCs. If they're abnormal shaped, if they have check the borders if the borders are not clean, if they have an abnormal color. Okay? The reason why is because if your cell is not contained, if your cell is not controlled based on these cell cycles, then the shape and the color and the borders will start being abnormally shaped. Okay? Normally a mole is very round, right? It's very round and brown. But if you start getting a mole that has not round, but it's almost looking like an amoeba, okay? Of course, you all know what an amoeba looks like, right? It looks like a dead mosquito on your windshield. and it's starting to turn like instead of a brown color, like a blue or a red color, then those are the ones you need to check. Because what happens is that these cancer cells will start to abnormally appear, and they will not be destroyed, and they'll start to grow not regularly. Okay, so everyone's freaking out now because y'all like these are lucky moles. But it is worth noticing these things. Okay, my brother was a goalkeeper for years and years and years, and so every day at practice he wouldn't be wearing a shirt, and so he's actually had several moles removed that were precancerous because of that. And so what he found was basically were moles that were abnormally shaped or different colors or stuff like that. So. Okay. So I hope that makes sense. Let's play a game.